diffusion enthusiasts. Okay, sounds like we're good. This meeting is being recorded.
Good. Thank you for the presentation, and I'm looking forward to give this talk here. So maybe we'll take a bit more than 40 minutes, but I will see how it goes, and otherwise just stop halfway. But I'm going to talk about the 3D organization of axons, and also I will try to link this to the structure-function relationship. So how does microstructure actually infer something to the function? And when I'm talking about function, then it's about the conduction velocity along axons. But we start from the beginning. So we have our T1 weighted MR scan here, and
I'm really sorry, Tim.
yeah.
You're not showing your screen at the moment.
Okay.
Sorry.
Maybe that's a good idea to do. So, like this, everything is good. I just need to get you out of here. Good. So we start here with the T1 weighted and look at the white matter. And here we know we have all these projections we have here in the corticospinal tract or between the two hemispheres. And we can zoom into these axons here. We can zoom in here. We have along here the axons where we have the white matter in the axons, the myelinated axons in the white matter. And then we have, of course, the terminals and the somas in the cortex where there's like the input-output of each neuron. We can zoom in to this to nanoscale because that is actually what we are measuring in our low resolution MR scans. And we see this bundle of axons, and when we cut through it, then we get this cross-sectional area that many of us know where we can quantify the size of the axons and look into the myelin. And from that, we can get this distribution of axon diameters. So why is it so interesting to look at these axon diameters? And that's simply because that these are actually the one that ensures the communication speed or moderate the communication speed between different functional regions in the brain. So if we have a situation where we need to run, for example, in this case, then it's pretty smart that our long connections here down to the legs, when we give the commando in the brain, add them actually to run fast and have a fast reactions. And this is one of the things, this speed is what these axons actually are modulating. There are many different features along the axon that actually control the speed here. And this is by Drexmet and Derek making this for some years ago. This nice study, which actually have different parameters that contribute to the conduction velocity along axons. And when you have a myelinated axon, then the diameter is one of the parameters that influences. But there are many other parameters that also influence on the conduction velocity. But we can draw it like in a simple way here in this cartoon here. So we have the axon and the myelin, and then we have conduction velocity along the axons here. And we can make this linear relationship that had been shown already by Hosch and colleagues and others before that, the larger the axons, the faster the conduction velocity is along the axon. What it also shows is that the myelin also is related to these conduction velocities. It changes also with the diameter. So what happens actually with when we have a disease, lack of myelin, for example, along the axon, that is something we really don't know much about. But it will change the conduction velocity when we have this structural change. So the formulation to go from the conduction velocity to predict it from the structure, and then also from measured predictions to the structure is actually a very simple formulation. And this is this conduction velocity where we have a constant factor here with a G ratio. So that's something that the myelin boosts. And then we have the diameter. And this is from the structure to estimate the conduction velocity. Then we could go to the conduction velocity where we can predict it from the latencies. So here we stimulate the neuron here, and then we have a signal travel along the axon, and we measure the conduction time. And when we know the length, then we can also calculate the conduction velocity. And in this way here, we can make a relation between this structure and function. So this is the structure-function relationship that I'm talking about. What we just need to know is something about the G ratio, which is the inner and outer diameter here. So actually the thickness of the myelin and diameter, and we need to predict the diameter. And what we also need to know is the length of the axon and then we need to measure here the conduction time. To get this insight into anatomical structures here, we can have different methods to measure these axon diameters. And this is a show here. So we have image resolution here, and then we have the features, so the size of the geometries. And what we can see here, this is the MRI. They have a low resolution, whereas other technologies, they can go to very high resolution. And they can go to so high resolution that we actually are in this sub-micrometer regime needed to see these diameters specifically that we also see with electron microscopy. So we can say in this range here, this is where we're mapping the organization of fibers. So that is from the big pathways down to the small fascicles. And then we have in this regime down here, then we have the more microstructural where we measure the sizes and geometries that we need to understand the structure-function relationship. So when we want to make the link between different modalities, then we have our traditional MRI setup here. And now I write 3D imaging because the thing is that the MRI is a 3D imaging technique. But what is important here is also that the structure we are measuring are in 3D. So we have a tissue here. We set up our acquisition. We have some knowledge. From that, we construct a biophysical model. We fit to the data, and then we can have a prediction, and we can apply it to something out here. So, but what is also important is that this knowledge that we have here, that is obtained from these histological slices. And traditionally, this is coming from 2D electron microscopy. And what I will talk about here in this talk is a specific focus on 3D X-ray synchrotron imaging. And I will also talk about electron microscopy that we use to measure the axon diameters related to the structure-function relationship. But before we start to rematch these different technologies, then we need to be aware of how we actually process the tissue. So we start with a brain in the MR scanner. That could be a human. Then we go into the dark side of life. So we take out the brain, we perfuse fixate it, and then we have it here in our bag, or how we actually do it is hydrated. And that's where we do the MR scanning. We do a lot of processings here if we want to validate it with, for example, the electron microscopy. And we stain the tissue to be able to see the myelin and so on and so on. And then we have the electron microscopy where we can do the imaging, or we can go to these synchrotron facilities. But what we need to be aware of is that when we do these different processings, then at some of these steps here, in this step here, when we start to prepare the tissue samples here for electron or synchrotron imaging, we do a dehydration step. So that means here that the tissue here has been dehydrated, and that means we have a shrinkage. And in the MRI, we have a hydrated tissue, so it does not have the same kind of shrinkage that it has in this regime. So that is what we need to be aware of when we want to do this comparison between modalities. What is also important is the quality of the tissue. So what we want to have is, ideally, we want to know everything in vivo, but that is not possible. So we need to do it ex vivo. And here it's super important that we have a very short post-mortem interval. So because it has been shown here that already after 6 hours, then you can actually see that the myelin layers start to loosen on the axons here. And if it's very long time here, then you can actually see it's completely dissolved. So this is very loosely myelin, and the cell membranes are also dissolved here. But if we do in animals, what we could do there is perfuse fixation. That means that we actually can fixate the tissue before it actually starts to discover its death. And that's the most ideal case where we have a post-mortem interval of 0. But when we start to compare things, then this is just examples how important this post-mortem interval is. So we can take some human samples. So this is something we have been doing with electron microscopy, with synchrotron imaging. So it's a 3D image volume here in 75 nanometer resolution of a white matter sample. And you can see here, there's a lot of white spaces here, which is something between tissue. And if we zoom into one of these slices, this is one of these slices here, we can see that we do see structures in white matter here, but we also see that they are a bit weird. So in this square here, we can see that the myelin here start to loosen. We get some like bleeps on the myelin, and it start to be very behaving strange. Sometimes it is dissolved. If you scroll through this 3D volume, then you will actually see that the myelin start to bleeps, And sometimes it actually start to not be appearing, and then it will appear again because it start to dissolve. So this kind of tissue, if you not have control of the postmortem interval, then you cannot trust it as a microstructure to evaluate.
Tim,
So yeah.
I'm so sorry to interrupt, but I understand there's at least one person in the waiting room, and you
Yeah.
have, if you just turn that off, I'm again really sorry, but I know they wanted to join.
Yes, I will also take control of that.
Sorry.
Uh, Waiting room. Mid all. Now, okay, good. Sorry. So this was this postmortem interval, which really matters. And these 60 hours are actually not, that's actually normal when you have old brains that have been stored for many years. So the procedure has to change. But if you handle the tissue in the right way, so this is a human brain with just 1 hours of postmortem interval, and we did an electron microscopy just to check it up. Then you can see here that the myelin sheets here, they are nicely wrapped around the axons, and you see a lot of nice axons here in this environment. But that said is that the diffusion MRI, the FA maps, they are still different. Why? I don't know. So what we need to be aware of when we go to this ex vivo part is really to check the tissue quality. Now, when we want to quantify this kind of microstructural features, then for many years, it has been said, that is before we started the diffusion axon diameters, that the axons, they are assumed to be cylinders. And this is an example of electron microscopy across corpus callosum, where we can see that it approximates somehow to cylinders. But what we also see is that when we see in some cases, we have some axons that actually are not projection in the same range, and therefore they, of course, will have a shape, which is because it has this oblique trajectory in the direction of the fibers. And that has been handled in a ways where it has been assumed that axons are cylinders, so you can take this simple matrix, elliptic minor axis, or equivalent diameter, where you say that this shape here is something because the axon was not oblique to the direction that we actually were measuring in our slice. But when we take some histology, then we can just acknowledge that the complexity of axons is not just straight. It can do this kind of undulations. You see it sometimes in the white matter, and you can also see that you have this kind of local crossings. So it's more complicated. So we want to see how these axons actually looks like in 3D. And this is where we go into this synchrotron facility. So synchrotron facility is an X-ray machine. It's a gigantic X-ray machine where the X-ray is so homogeneous that you actually can see the finest details here. So it's actually a nanoscope that you can do. And this is the one in France, Grenoble in France, and this is the beam station here that we are acquiring the data. So this is down here, the optic is that we are using. So we have our brain sample that we have scanned, of course, with MRI, and you take out these small punctures tissues here. And then you prepare them with osmium staining and embed them in an EPON. And then you put them on a sample holder like this one. And then you go into your machinery, and this is the beamline ID16A in Grenoble. It looks like a space station. So you have the samples here, and then you put it into the synchrotron here, and the beamline will come through in this area down here. And when you put the samples in, then there's a robot arm taking your sample and place it down in this vacuum chamber before just a collection of data. Collecting of this data takes about 2 hours to get that. And what you get out now is this data set. So this is example. So this is the surface of just below cortex. You can see the cells, a big blood vessels coming in, and you can see the axons. So this is a crossing fiber in the cortical spinal tract in deep white matter. We can see a lot of crossing fibers. And this is corpus callosum, where we see a mostly straight fibers here. So this is this 3D volume. So we do no cutting or anything. It's like an MR scan. We can scan the whole intact volume to see these 3D images here. So these are in isotropic 75 nanometers. So what we get out here is this volume here. And we look at it here, we can see this blood vessels here. We can see the axons coming up. We get vacuoles, cell clusters here. The thing is with these vacuoles, they are these high intensities. We don't know what it is. Actually, we think some of them are related to a tissue processing artifact. Some describe them, and other times we don't see them. So they are like what we call the unknown. Then we have the cluster for cells. They can be glial cells that are put in here. And you can see if you do a Nissl stain, you will see the same kind of clusters here when you do in Nissl stain. So these are some of these features that we can get out. And of course, we have this nice 3D data set, and we were so happy when we got it out. And then the big question is, how do we actually process it? It's a nightmare to get started. This is big data. And that's why you start to have this naive PhD student. This was Mayam at that time. Now she, of course, grown and be less naive, so she know how complex this is. But we started with segmenting these cells here because they are pretty easy. That's just round optics. So you can have the cell clusters, the blood vessels. They are so big so you can easily detect them. And then we have the vacuoles, which are also round. By segmenting this with like heuristic methods, a combination of different standard methods, then you can actually extract all these features here. And we stack 4 volume on top of each other, so we get like a volume that is the same length like an MR voxel. And then you can start to quantify the statistics, the sizes, and the distributions of these features here in 3D. And this information you can use to generate synthetic data or just to understand how things looks like. But the challenging part is to segmenting is actually these axons here, because they are long structures projecting through the whole structure or the whole volume. So the first solution we did was simply to crop out, because the data set was so big, so we could not handle it. So we crop out each axon. So we identified roughly with a center line, and then we took out this piece of axon, and then we did, we did this analysis, surface layer analysis. So we extracted set spokes, so we could analyze where do we have the myelin, outer myelin layers, and where do we have the cell or the cell membrane of the axon. And then we have this layered here that is easy to analyze the sizes. So, but there's a lot of manual work to really realize this. So in the end, we ended up Mayam segmenting of these axons here. So this is 56 axons of the largest one that actually could be segmented manually. And what we see here, this is in corpus callosum axons. The colors represent the axon diameter variations along this one here. And you can see these are not straight, and they have a lot of axon diameter variation along the trajectory. But the cool thing is now when we have these 3D structures, we can start and actually quantify what is going on, so we can get some statistical knowledge about these structures here. So overall, we can see we have a distribution of axon diameters here. This is for all of these 56 axons. And you can see they are all about 2.5 micrometer, which is just indicating that this range of diameters we took out. If you take one of the bigger axons, this is the largest one, then that has more variation than the smaller ones. And this trend we also can see with the newest segmentation methods we are applying. The larger one have more variation than the smaller ones. But what is important is that we also explored that this axon diameter, this distribution of axon diameters in 3D, actually, when you do the same analysis in 2D slices, that what we know from traditional histology actually show to have the similar axon distribution. And that is important because then we not always need to have the 3D structures to do a quantification of these sizes here. But what makes these sizes is interesting. So why do we have this variation? So if you look at the blood vessels here, then we can see that these are like pushing to the axons. They go around here. And we can see that this changes the axon diameters. We can also see we have these vacuoles here and the glial cells here actually pushing to the axons. So the axons are like the soft material in the environment. So it's an extra-axonal environment that push to these axons here that are contributing to these axon diameter variations we have. And now we just look at the static image. But actually, there's this nice study here by Tönnes and al. So this is in hippocampus. So they make a small lesion at the yellow cross here. And then you can see here after a few times, so you can see the time is running. And these are the glial cells coming in and inspecting these lesions that are in the hippocampus. And what we see here is that these are just moving around in the extracellular space and just pushing to these axons, and these will change their shape because something needs to be soft enough to allow this kind of dynamics in the extracellular environment. So this can explain why we have this kind of diameter variations in of the axons. So when we have this long structures here, so we can stack together these axons into 4 volumes. So we can also start to analyze the trajectory of the longer one. So this is one axon which is 660 micrometers long, of the same going through 4 of these image volume that have been stitched together. And what we see here in the stars, this is the node of Renoir, and they have a distance approximately about 300 micrometers. We can zoom into this one here and look at this. And what we have at the node of Renoir, that is where we actually have one of the thinnest parts of the axons here. So the axons have a size, and then at the node of Renoir, they become very thin. And then that part here, they also do not have any myelin, as what we expect. And if you map out here, this is the length of the axon, and you look at the axon diameter variation here, this is the blue, and then you have the G ratio, that is the orange here. And here, at these points here, here, then we have this point here. These are where we have the node of Renoir, where we have the smallest diameters we can see that we have here. But we also have another example is these vacuoles, these unknown that we really don't know what it is. But they are like forming up also, forming up around the axons and sometimes. And you can see them pushing them down to be very small structures here, diameters. And this is a 3D reconstruction of a vacuole that are like pushing to the, or squeezing like the axon in this local region here. What we also interesting find is because we can measure like the axon diameter and the myelin thickness along these long trajectories. What is interesting is that the G ratio here, which is a ratio between the inner diameter and the outer diameter, the myelin variation, more or less follow the trend of the axon diameters. The myelin thickness is constant along the axons that we have. So that suggests maybe, that the myelin thickness does not need to change so much as one might think for the traditional way of thinking of G ratios. So this was really zooming into the individual axons in the very fine detail. But we want to look at the structure-function relationship, and we want to look into a population of axons. We need also to zoom out a bit to look at how things are organized. And this we also did here. So this is a recent study that we published in eLife, where we were looking at the MR scanner. We have the same pixel we have the same sample from the MR scanner scanned at 2 different synchrotron facilities. So at the DAISY here, this is the one in Germany, in Hamburg. Then we have a 500 nanometer resolution. And then we go to the France, the example I showed you just before. So this is the same sample here. We go down to 100 nanometers or 75 nanometers. So what you see here is that to get the contrast to myelin, then we use, like everybody else, osmium staining. But the problem with osmium staining is that it has really a low penetration depth. So if we have a sample here, this sample here is around 0.8 millimeter, then you can see there's a very thin rim here that the myelin stained, and the rest of the sample in the center, it did not penetrate it. So that's why it appears non-stained here, it appeared transparent. So when we compare here, so we have the different sizes. So this is an MR voxels squared. This is the sample that we took out and scanned it. This is the field of view of the DAISY, which have covered the MR voxel, and then we have the ESF at the highest resolution. When we stack together the different volumes, then we can actually get one length that is at least matching the MR voxel. So when we look at this structure here, so we start with the tensor imaging here, diffusion tensor. So this is the color coding in the corpus callosum of the monkey. And the white spot here, that is where we took out the sample. So this is the red, and then we have the second is the blue, and then we have the third eigenvector direction here, that is the green. And the FA is high as expected, and you can also calculate the micro FA here, which was just a simple multi-shell with cadence SMT method here. And when we look at this and compare to the synchrotron imaging here, so we applied to the synchrotron dataset structure tensor analysis. So the structure tensor somehow also have a tensor shape that we somehow try to see if we can link with the same features that we can extract with the diffusion tensor setup. So we apply the structure tensor here, both to the DAISY data with a large field of view and then to the high resolution data. What is interesting is that here, even in the region here of corpus callosum, then we can see the color coding here is blue and a bit of yellowish. So here we have a different fiber encoding. So the fiber encoding is actually along the fiber. So we can see that we actually have some laminars of sheets here of axons. So there's some which are yellow, and then there's some blue, and then we have yellow, and then we have blue. So that means we actually have along the axon in this region here, we have laminars that have like an angle like this. So they go in the same direction, but they have a intersection about 35 degrees. When we look at the high resolution data set here, because it's a small volume, unfortunately we cannot make exact match here. It's the same sample we had acquired, but we cannot find the same match at this data here from the synchrotron. That did not succeed, but it is in the same sample. And because this one here is smaller field of view, then you are not guaranteed to see the same kind of laminar structures that are seen in this larger field of view, simply because this is small. So when we are comparing structures here with diffusion MRI with a relatively large voxel, and we say, oh, this is super cool, we can do this high resolution, but if our field of view does not cover the same voxel size that in the MR, then we'll risk to have a mismatch here and lose some of the information that we clearly see here in this case. We can also compare the anisotropy with structure tensor. So for the structure tensor, we used a scale space approach. So the pad sizes, which look at the neighborhood of the structures. So structure tensor looks in the images for stripes, which have like the same direction. And the information of these stripes is something that we expect to come from the cell membrane or the myelin from the axons and whatever is in this environment, which we expect to be something that the diffusion MRI also detect. So we can actually look at the diffusion tensor also in this microstructure or the structure tensor with the shape in this environment too. And if we optimize it for anisotropy, then the highest anisotropy we can get is about 0.75 in this data set here with a high resolution. And we can see it's a very high dense, or we have it pretty high FF values here. And we can see when we go to DAISY, then this functions is like spreading out. And that's simply because that we now start to go to a lower resolution and the features start to be blurred out, which are not what which is of course a natural case when you go to lower resolution. So when we now, we can also go to mice here, and we do the same experiment. So this is a mice and with the corpus callosum we have here. And when we look just on the diffusion tensor, so we have the corpus callosum here, red, the principal direction, and then we have green and blue. When we look at the structure tensor in the same region, we get the same information like what we get in the diffusion tensor. But what is interesting is that we actually have a different organization here. I just need to have somebody in the meeting room. If we look at the monkey brain here, if you just zoom back here, then we can see we have blue and we have green, and here we have green and we have blue. So that's an indication from the other principal directions that are some different in the organization. And here we do not find any kind of laminar organization like we did find in the monkey brain. But when we look in this study here, so this is Su et al. So what they did in the mouse brain, they injected a fluorescence in the M1 and the sensorial cortex here with two different colors. And when you zoom in and see in the corpus callosum here, you can see the hair are stacked on top of each other. So it's two parallel bundles that are mapping on top of each other. So that agree why we don't see any laminar organization here, because they're simple parallel. What we also did have a try here is to see how is this structural tensor actually working if we have an environment where we introduce a focal lesion. So this is a Krupperson mouse model, and you can see it develop a lesion here, demyelinated lesion in this region here. So we can see the myelin from the Osman staining, and then we see the lesion area here. And this is a healthy control. And we can see that here that the fibers are going throughout the whole corpus callosum, or this part here. But interesting is that we also, in the area of the lesion, we still have the sensitivity to the directionality of the fibers. And of course, we expect that because we just lose the myelin, but the fibers is still penetrating throughout the fibers. And it can be shown that we also can detect, when you look at the shape of the tensor of the anisotropy, you can see it changes in the structured tensor. So this was in corpus callosum. So what is actually happening when we go to more crossing fibers regions? So this is in the monkey. This is deep white matter in the centrum semovalar. So this is this fiber where we approximately took out the samples here. When we look at the spherical deconvolution, this is FOD we collected out here. And we have the FA value here, which is low because it's a crossing fiber. And the FA, the macro FA, is high because it's not sensitive to organization. When we compare this FOD here with the one in the structured tensor here, then we can see they are alike. There are bit differences, and that is simply because that we do not cover the whole field of view like in a voxel. But what is interesting is here, when we see this color code here, like RGB colors, like in the DTI, then we can see here that we have like layers, laminars of fibers stacked together. So we have blue coming in here, then we have a layer of red coming out here, and then we have a blue, and that is like stacked on top of each other going through this volume here. And we also have another dimension here, that is the green, that is a bit hard to see because it's coming in the different directions. So they are coming in here, and they actually crosses more or less on a grid wise. We can also analyze the anisotropy because we have the tensor here, and these structured tensors, when we took at the anisotropy, we still have the same high anisotropy like we have in corpus callosum. So that means, and that's also not surprising, that we are in such a high resolution here that we don't detect the crossing fibers. Actually, we are at the same, the scale space actually has the same size approximately to the diffusion propagation distance for this setup we have acquired. So this agrees me here, low FA values because we are sensitive to crossing fibers. Here we have such a high resolution that we don't see the crossing fibers, but it's more in the same regime that we get with this micro FA. And just to have a visualization of how things looks like. So these are this volume is just one volume, but we can stitch it together to make it much longer. And then we can play this movie here and then scroll through it. And then you can actually have a feeling of how these laminas, looking at the color, how they're projecting through this large field of view here. So that's super cool, if you ask me, but I'm also a bit biased. Okay, so what? The thing is here that this also triggered this discussion here that Van Verdien actually mentioned here in his science paper in 2012. He came up with this tractography that we have. These big tracks here seems to actually interact on it across each other like on a grid. And he showed for different tracks here in the monkey. And there was like a discussion about this. This is Cantani and colleagues showing that this could actually just be an artifact to tractography because of the low resolution. Then Van Verdien replied again and said, but this has actually been shown in old storybooks where you can see the fibers coming in this specific, in this deep white matter, deep centrum semoval, where we are crossing fiber, that you have this grid crossing of the fibers here, and come with some examples that you can see and also indicating by tracers. And what we find is here is actually that this angle is not just a grid or not a grid. So it seems that when fibers are crossing, that we also see in this region here, which is the same here, then fiber crossing, they more or less always cross in a high angle. So that's about 90 degrees. If there's two fibers along, like what here in the corpus callosum, then they can have different they can have a low angle. So that is from 0 if they are completely parallel, then we cannot see it, but they can also have up to a 35 degree or something like that. And this also agrees what we can see here with this parallel organization of fibers that are still laminars of these fibers that are following each other. So, and this is also something that Van Dien actually mentioned at his paper, is this organization of a grid that I think is laminar, and also the low angle and the high angle actually come from the early stage of the development of the axons. And this is actually something that has been shown here with Smith and al. Show this nice story. So here they have a population of axons, and you can see all these stripes. This is the growing of the axons. And when we apply this, so this is after 12 hours. And what you see here is that first you have axons like spread out, and then they start to collect together, like into bundles. What is happening here is what they call the zippering or unzippering effect. So this is the effect of fasciculation or defasciculation. So if the axons are follow the same direction, then will at the beginning where they have the growth cones and have been stable, they will try to form a bundle. And this is coming from a mechanical and a chemical process. But what is cool here is actually that this process also work when you have not only bundles, but also if you are crossing. So if you have a crossing, then this mechanical forces and the chemical forces actually changes. So if two fibers want to go opposite direction, then they will cross, and they will cross on a grid line. They can also do unzippering, so two fiber balls can follow, and then they can unzipper and follow up. And we see that from also the major bundles that they follow in the IFOS, and then when they project into the cortical regions, they spread out in minor tracks. So it indicates here that we have this laminar organization, and it is something that are bundled, and it has something with different inclination angles. And what they say also in this paper, which is interesting, is that this effect will disappear when you have the myelinating effect. So it's only in the very early phases this happens. Okay, getting back. So this was the organization. Now we get back to the structure-function relationship. So to look at this, how structure infer on the structure, on the function. Then we did this study here where we compared in the rat brain electrophysiology, and then we did electron microscopy to see with EM, and we did axon diameter measurement with MRI, and we did tractography, and all these things here that allow us to see how different modalities actually agree on the prediction of electrophysiology on the latencies. And just to remind you, the idea is that to be able to predict the latencies, we need to know the diameter of the axons, the G ratio, and then from the latencies, we can predict the corresponding conduction velocities if we know the length, and we know the time it took, the conduction time it takes from stimulation to out here. So the setup is like this. So we have a rat brain, and then we injected a AAV virus, optogenetic virus. So when you light on these neurons in this area here, then they will actually start to fire. But its AAV virus also carry a tracer, so you can actually see which levels, which axons actually are stimulated. So when we do it, when we look at this here in the corpus callosum, so we injected here in M1, and then you can see here weekly the frozen signal here. This is the bundle, and what is interesting is that this, you can see here is on the top. This bundle here is only seen on the top of the axon, and this is what we also saw before, that the M1 is a laminar that will be put on top of on the other projections. To get the distance on ex vivo, we collected the tractography. And with the function, we stimulated here with a laser, and then we see here the conduction velocity, the latencies. So we stimulate, and this first signal here, the P1, which is the evoked LFP signal, that is the one that we actually use to predict the conduction time from here to here. You can have many timings here. This is just to show, this is the P1 peak in all these animals we did. And you can look at the different time points here, how they develop over time. And this is just a checking that some animals is not just jumping in time completely crazy, but it's just this is a nice homogeneous way that they follow the different timing when they're stimulated. So this is the latency we're measuring. Then we have the axon diameters. So we have the standard EM that we all, or many of us, have used for many years. And you can see here we don't have any extracellular space. But if we did instead in the same animal, in the same region, no, it's not the same animal, it's a different animal, but in the same region, we do cryo-EM. So cryo-EM is that you take the fresh tissue and then you high pressure freeze it immediately. So after a few milliseconds, it's completely fixed. And then you stain it with osmium like you do in standard EM, and you can embed it, and then you can image it. And you can see here we have a lot of more extracellular space here, and the shape is changed. So something is changing. So we have some effect of tissue shrinkage. So if we analyze this, we have the standard EM. We have the distribution here of axon diameter, and then we have a cryo-fixed here, and we also have the axon diameter distribution. So if we look at the peak here, the mode, then we can see that the mode here of the cryo-fix is a bit larger than the one of the standard EM, which indicating fixation. But when we look at the axis here, so this is 1 micrometer and this is 1 micrometer, we can see in the cryo-EM that we have many more axons here that are larger than what we see here. So it's so this suggests that the larger axons are shrinking more than the smaller ones. And actually, we found it that it's actually going up to 48%, and the smaller one is about 33%. And we can have if we look at this is a shoulder that we have like here. It's a simple way of marking between the two relations. So it's a tail here. Then we have this shrinkage factor, which is much larger for the larger axons. Then we have the diffusion MRI, what we have done. So what we know about this, that it has a sensitivity profile, and this sensitivity profile determines the range of diameters that we can measure. This is shown for many years ago with the voxel domain, which we did for many years ago. It's just showing here that the smallest diameter up to about 3 micrometers, we don't have any contrast to the axon diameters. But for the larger one, we start to see, if you increase the gradient strength, then you start to see much more clear the distributions. And it's the same for the models we're using today, these micro domain tensors here. But they still have the same kind of axon diameter profile. So this is the true diameters, and this is predicted. This is ex vivo, and this is the in vivo connectome with the predictions, where we can see we have this lower axon diameter bound, where below this one here, we don't detect the axon diameters. So we only detect the big axons here with this setup. So now we take these all these different modalities, and then we put it all together to this structure-function relationship. Because we have this simple linear relationship, then we can actually here on the x-axis say the conduction time in milliseconds, and we can have the scaling here to the axon diameters. So here we have the electrophysiology. We have the EPON with the axon diameters, and then we have a cryo-EM, and then we have the diffusion MRI. So if we start to compare here, so we have, and this is mainly done of all the same animals except for the cryo-EM. So if we look at the structure-function relationship here, so these are the all the animals we did have has here a conduction velocity about these 7 milliseconds here, and that corresponds here to the mode of the electron microscopy. So if you account for, so this is the distribution here does not account for the shrinkage, but if you account for the shrinkage that we have learned here for the cryo-EM, that is like the stripes here, that is how we shift the mode here, then we can see that the electrophysiology actually agree still with the small axons just below the mode, what we see here. Oops. And if we have, that is the correspondence of this one here. And these are the, if we have the corrected, as I said here, with the cryo-EM, it's still the small axons that we have. And if we correct here for the shrinkage factor here with the electron microscopy, then it actually somehow agrees what we see with the cryo-EM. And we used a general linear scaling of 40%, which does not account for this nonlinear that we actually found. Then when we look at the axon diameters here, what we find here is that we have the mean diameter distribution here in corpus callosum, what we have here. We can see here that it fits here to the tail of the axon diameters. But actually, what we did here is that because these axon diameters, they are volume weighted, then we also take the cryo-EM here and volume weighted it, so it shifts the distribution toward this direction here. So, but it's just indicate here that the diffusion MRI, as we know, is sensitive to the larger axons, does not detect, or what we can say, the correlation here with the electrophysiology here does not detect the distribution of diameters that we actually are measuring with diffusion MRI here, because they are in a different regime than what this setup with the LFP signal actually is detecting. So different of these modalities here has different sensitivities to the structure, or they see that the structure's function relationship with different sensitivity profiles. So we can put it all together. So we can compare across modalities. So here it's important that we account for this tissue processing that we do. It's all these modalities acquiring the tissue in different states, and that's would bias it across these measurements here. One of these, what we found, is the shrinkage factor when you do EM, which is nonlinear, and then we also found out, or I did not show any result with the postmortem because that's waste of time. So we just need to be sure that the postmortem interval is as short as possible, so the microstructure environment is something that looks like in vivo. Then we have here synchrotron imaging benefits to offer a large field of view. So we can go up to what, so we can actually map the size of the axons. And you can apply this structure tensor analysis of this synchrotron data here, and then you actually can get something that somehow can relate to what we see with MRI in the same range. What we also saw with the synchrotron imaging is that we have this laminar organization, and that changes also across anatomical resolutions. So we can have big fiber bundles, and we can even those that can be seen with the major pathways that we can see with diffusion MRI. We can go to smaller ones and down to the smallest fascicles. They all seem to have this kind of laminar organization. And then we observe this crossing. So if we have a low inclination, this is not right. This should be high inclination. It should be grid line. Then we have crossings. If you have fibers going fanning or parallel, then they have a low inclination angle. So then we have also, we can do the quantification of axon diameters. So one thing is that is important is the synchrotron can show this 3D histogram of axon diameters. And these histograms here can be compared to the histogram, 2D histogram that we do, for example, with EM. One thing is that I did not talk about is the synchrotron here has a limited resolution. So there are some features that you cannot see, like the smallest one, and that's where you can combine the information here with the 2D histogram with EM that can go to higher resolutions. We need, of course, to account for this shrinkage factor here that is happening. And if we do the shrinkage factor, then we approach more the EM, EPON, the traditional way, to match it with the high pressure freezing that is like the way of doing, so we can see the extracellular space. But still, we lose information here. Then, of course, also the axon diameters is not the same kind of diameters that we're measuring with EM. They're volume weighting, and these kind of things we need also to account for. Then finally, the structure-function relationship of conduction velocities of long axons. The different methods have different sensitivity profiles, and that is something we need to be aware of when we try to combine this information, or we combine structure measures with the literature, with electrophysiologicals, especially the LFP signal that are often used, that relates to the population of small axons. So this acknowledgement, thank you. So many people have contributed to the studies here. And this is many in this group here have been joined, and also my previous PhD student, Christian Skoven, doing all optodynamic stimulations and some collaborators, and of course a lot of fundings, and been funded also for my ESC consolidated grants. Thank you.
Thank you so much, Tim. Let's, if you could stop sharing your screen, 5 minutes countdown, 5 seconds countdown, everybody, please. Turn your camera on so Tim can see the audience. It's about 3 seconds left. There you go. And now we can give you a round of applause. Thank you so much, Tim.
Thank you. Oh, I'm sorry, I see more waiting room. It's hard multitasking here.
Well, you've got the most good-looking people on the call anyway.
I can see it. Thank you very much.
That's the thing that matters. Okay, as I said, we give the first question to a trainee, and our trainee is Bob. Don't be nervous, Bob, if you'd like to start off with the first question.
Not exactly a trainee anymore. Thanks very much, Tim. It was a really, really good talk, really wonderful stuff. So nice to see that Van Wedeen was right about several things. I've got a question about the corpus callosum and the laminar structure. Chris Wiggins and us, we published a paper in 2017 showing that we could see very high resolution structural MRI. We could actually see a foliate or laminar structure in the human corpus callosum. Mm-hmm. Do you know about this paper? And do you think that relates to... Okay, well, I should send you a copy then.
Please do that. Yeah, I've been looking for this. Please do. I also see Amy's. I've been looking very much into Amy's data also to see if we can reveal it, and in the polarized light images.
Well, you can look up Chris Wiggins and Robert Turner. You'll get it. Radiology or some such channel.
Thanks, Bob. Now, Amy, and that was not a real phone number in the chat. Over to Amy.
No, I Googled that phone number and it comes up with something very weird, so don't contact it. Fantastic talk and like a huge amount of work. Also staying on the corpus callosum trend. So at least in our data, we see, really interesting patterns of like kind of wave-like patterns that I think are all related to this, but in a kind of 2D sense, but that can change across the corpus callosum. So like different things at the midline and different bits not at the midline either side, and that probably changes as you go front to back. And I was wondering, have you got data that's going across the corpus callosum? How consistent do you think this is? Like, I think in the middle that you show that actually the colors of the second and third axis of the tensor flip. Like, do you think the laminar structure could be kind of twisting, or like, how do you envisage this structure kind of going across between the two hemispheres, and can you relate that also to kind of formation and development? And anyway,
that was a very big question, Amy. So
sorry.
So actually we did not measure exactly in the mid-sagittarial corpus callosum. It was a bit offline.
Yeah.
Compared it also with different HPC human datasets just to check that it's the same. And we do see the same patterns. It could be very interesting to see, because I think that the fibers in the corpus callosum are coming out in like sheets, and then they go turning like this in the mid-sagittarial, and then they start to go like this. And what we are measuring is this when they start to fan out very weakly. And that it could be really, really cool to be able to show that. But I think the paper with Bob, maybe you can see these structures. You can also see this with tracer studies. Giorgio Innocenti have been showing some straters, but you also can see this columnar organization. So that is well known that it's in there in the corpus callosum. But I don't know if anybody have mapped it going through. So that could be super cool.
Thank you.
Thanks, Amy. And I've put the link to the paper Bob was referring to in the chat. Any other questions, folks? All right, well, maybe I could start with one, Tim, and it's just very simple. On the color DTI maps, the second and third eigenvector, I naively, having never really looked at the color, assumed they'd be fairly random, perpendicular to the orientation of the, you've got the red for the main direction, but it was uniformly green and uniformly blue.
Yes, that is absolutely. But nobody have really been looking at this at the detail on the other directions. It's completely uniform. It's not random at all. And what surprised us was really this change in the second and the third. And I've also checked it in rats. So it seems that rodents have this different kind of organization, whereas the primates and the human, they share the same kind of information. And in the paper, in the eLife paper, we're also speculating about this, that it is a very simple metric. Sorry, I'm still letting people in. It's a very simple metric. It's a very simple metric to say something about the organization that we have not been thinking about until now. So maybe we should dig into this simple metric.
Yeah. No, it was cool. I mean, I'll give you all the compliments at the end, and I had lots of cool things to ask you. But I see that Matteo is waiting, so I'm going to hand to Matteo.
Hi, Tim. Thanks for the extensive talk. It was very interesting. I have two questions. The first one probably is quicker than the second. So I've seen that for the structure functional relationship, you have stuck with the first peak, the P1 positive peak. If we stick with the single axon approximation, and given that we measure the larger axons, so the faster ones, have you considered using instead the onset of the peak, although it may be trickier to identify? And the other question was, in the final picture where you had a lot of things condensed there, I kind of missed if there was like some kind of scatter plot that showed what was the relationship between the delay and the axon from one of the modalities.
So yeah. So we first question first. That was the single axons with the peak. So we use the LFP signal. We tried. You can see in the LFP it's like silence period, and then it starts to increase. And it was very hard with this LFP signal to get so close down to the beginning of the pulse, where you actually see the first one start to firing. And that is simply just because that I don't think that the LFP is the right measuring technique if you want to push it. Then you need to do to the single cells where you actually, or you can have a multi-array where you inject, and so you can actually get more specific information when the time arrives on the individual axons. And that is just how it is. So that it's mostly so it sees something, but what we're measuring is the statistically most robust time is the top of the P1. The second question, could you tell me what the second question was? Then I can tell you the answer.
Yeah, so in the very last picture, I missed something like a scatter plot showing the actual relationship between delays and axons.
Yeah, we tried it. And that's because it is so hard to have the correlation plot. So actually the plot I showed where you have the conduction velocity and you have the diameters below on the axis, that is basically a scatter plot. That's the closest you can come to a scatter plot comparing different modalities. So we did try all these different things here. So, but yeah, but it could be cool to do it in a scatter, but it did not work out. We would be more confused.
Okay, thanks.
Thanks, Tim. Ali, please.
I think.
Hello, Ali.
Thank you so much for the talk. Great work. So part of the presentation, you mentioned that the changes in the larger axons were dominant compared to the axons with smaller diameter. That goes a bit against my intuition that I thought that perhaps smaller axons actually undergo larger changes.
In relation to the
did you normalize it by the, so are you looking at the changes, or did you normalize it by the mean diameter?
No, it's related to the actual changes. So the variance along the axon, so that we see. And to me it makes sense because that if you look at the axons, the bigger the axons, then to have more conduction velocity, then you also need to, in the intercellular space of the axons, there are more free space to actually send the ions along the axons. that means that this space will not be completely occupied by the cytoskeleton. So if you have something changes, the bigger axons will be the one that will be changing more in the diameter. They will be easier to change than the smaller ones. One thing is of course what we do not see in this case here, in this data set, we do not see the mitochondria. So for the smallest diameters, then you can see these bulks of the mitochondria. And I think you have shown it in one of your EM for the axons. Then you can clearly see them. You have an axon and then there come a bulk with the mitochondria. We don't see that in this data set here, but we see it in human data sets.
Yeah, because for the smaller axons, the size of mitochondria is really close to the diameter of the axon itself, and when they are passing along the axon, they really change. I mean, up to a factor of 2. Then I was expecting to see the relation actually in receptacle.
That's true. Yeah, that's true. Yeah,
thanks so much.
Just on that topic, sorry, Dimitri, I do see you, Dimitri, but just quickly wanted to ask. It's a naive question, but do you know what comes first in terms of sort of imagine that if the axons grow to sort of trying to avoid the vasculature, because I think your argument was that a lot of the bends, the variations were due to the pressure of the vasculature. But is it that the axons grow first and then the vasculature sort of fits around it? Because you could imagine it's harder to fit a big axon between the vasculature, and it's likely to be more, it's harder to do that than if you've got tiny little axons.
Yeah, that's a good question. I think it's something that is over time. So when the brain grows and the vascular system grows, they all somehow grow together. It's and the axons are very. Have you ever stuck your finger down into the brain? It is like it's completely stuff. Yeah, you should try it.
Okay.
That is really interesting. So it's a very flexible structure. And that's also why we can hand it when we get this concussion, so you don't completely smash the whole brain network, because it is so flexible.
Okay, thanks. I saw a physical hand from Dimitri, then Bob.
So just on the topic of mitochondria, so were not able to resolve them with this X-ray, or why are you not really correlating them?
Because we did not see them. We could not really see them in a robust way in the monkey data set. I don't know, because they are there. But we
yeah.
So maybe there's something with the tissue processing or something like that. We don't see them. We also don't see them in the smallest one, and that could also be because of the quality of the data. But it's not as clearly as, for example, in some of Ali's electron data set. But we see it in human data set. Then we have a data set where you actually can see the mitochondria.
Yeah, so the first data set, just for reference, was from Hongxi Li from our group in 2020, and he correlated the mitochondria positions with the beads on axons. That's Nature Communication.
You did study about this. So
so that, and then in Ali's data set we also see that, yes, but and somehow that scales on which beading happens, a few microns. That kind of, I think, is more consistent with mitochondria, because they're pretty often, pretty dense along, rather than some vasculature that kind of happens every now and then, right? That the spatial positions of beads, the distance between them, like 3 or 5 microns, may be too short if it's all because of some vessels nearby. I don't know. Maybe Bob Turner knows this better.
I cannot answer because we do not see them, so I don't know. So, but I know that the structures. So, the general rule is whatever so you have the axon diameters is a flexible structure, and it's an environment around that changes it, if it's intercellular or is it like something around.
Yes, and I think you probably didn't mention also the work by Marios Georgiadis, where he did the X-ray scattering and correlated the ODF with myelin small angle X-ray scattering and correlated with diffusion MRI 2021. My actual question is about your microFA values. We know that microFA is bounded by 1, from 0 to 1, and you were showing values 0.99.
Yeah.
That's presumably unphysical, right? So what do you think are the biases for this value?
I think it's because it's a simple model. It's the SMT by Caden, and that
But that doesn't measure microFA.
no, but that is the point was it is a microanisotropic matrix, but it's coming out. But the main point I would show is that when you are insensitive to the organization of fibers, that is the only point. You get an anisotropic measurement that's related to this, and when you do the synchrotron imaging, then you can see you have such a high image resolution that you are not sensitive to the organization of the axons, and that was the point I would try to do there. It should not be interpreted as a precise estimate.
Because I thought this SMT measures the axonal volume fraction, but it's also between 0 and 1, right? And it's not, can't really be 1.
Yeah, I agree. I agree, Dimitri. So,
all right.
I agree.
Okay, thanks, Dimitri. Over to Bob and then Matt, please.
All right. This is actually a question that Dimitri has discussed with in the past, which is the distribution of axon diameters. The question is, why are there so many small axons? You'd think that that it would be ideal for the conduction velocity in a fascicle, in a bundle, to be constant across it, so that the signals coming from one brain area going to another brain area would all be arriving at the same time. So is there a distribution of axonal sizes? Because in terms of getting the message across from one brain area to another, you actually want a distribution of conduction times.
Yeah.
Or is there a distribution of sizes simply because by the time the smaller axons are developing and becoming serviceable, which is maybe later on in development, there isn't much room for them to expand in diameter. So they're stuck because it's a space-filling problem.
That I maybe. I think we should see it from a different perspective, because one thing we don't see when we do this kind of analysis is we don't see what type of axon what type of neuron this axon actually belongs to. So we have a whole higher hierarchical level where we have different type of chemical systems that also have an axon that are communicating. So maybe this distribution of axon diameters that of course is to secure the conduction velocity, the delays for the different brain system which is needed. And then it's also maybe related to this different chemical systems. One thing is sure is that the brain will never have too many big axons because they are super expensive of energy. So they only have big axons if it's needed. And if you compare, look at the mouse brain, the distribution of the mouse brain in the axons. First, there are many non-myelinated axons, and then there's also the distribution of the tail is very narrow because the brain is small, so it don't need to have this fast speed. So the brain will always try to optimize, of course, also its size. If you go to the rat brain, then you see the distribution start to change, but the main mode persists. And if you go to monkeys and humans, then you see that this, again, the mode stay, but it's a change of the large axon that actually changes across the species. So there are something, and the bigger axons only come if it's needed.
Well, I agree with that. Thank you. There's much more to be said about this, but we haven't got time. No.
Yeah, I would love to discuss it.
Yeah.
Matt, then Denis, please. Hey, Denis.
Yes, Tim, fantastic work. One comment is the beauty of biology, it doesn't care what we think should happen. It's what actually happens. But my question is related to your cryo-EM and the drastic difference between the traditional fixation methods and the cryo-EM. In particularly, it looked like your G ratios are very different. Were you able to image that in 3D and look at diameter distributions. Can you just elaborate a little bit more on that? And I guess maybe I've always wanted to do the high pressure freezing and rehydrate and image the tissue. Is that even possible?
So the heat rehydrating after cryofixation, that's not possible.
Okay.
But maybe you can ask the experts. The high pressure freezing is a very complex setup where you really need to have a machinery that can make this pressure, and the tissue needs to be prepared in the right way to make it actually to work. But I don't know much more about this.
Yeah. I mean, my understanding is it's under high pressure and it's still fixation, but it permeates very slowly into the tissue under freezing, right?
Yeah, so the whole process takes a few milliseconds.
Oh.
And then you have the high pressure, and you have a build up of high pressure I think it's 5 milliseconds you have it, and then it's high pressure freeze. Then it's in this cryo state. And then you take out your sample and put them in cryo, and then you keep it down there until it's stabilized. And then you start to do the staining and process the tissue and cut it.
Gotcha. Okay.
But the thing is with the cryo is that you can only do it in very thin slices. It's about 100 micrometer, because you need to secure that the high pressure actually cryo freeze the whole sample.
Okay.
So regarding the big changes we see, I could talk for 1 more hour about this, but there's a paper by a group in Switzerland, I can't remember. They explored the shrinkage factor in cortex, and there you clearly see they focus on the extracellular space, and that is massively shrunk in cortex. And they do this nice story where they compare the traditional EM with the cryo-EM. I do not recall that anybody have done this in 3D, and one of the challenges is that it's very thin slices we have. So I don't know how it looks like in 3D, to be honest.
Okay. And the G ratios, I mean, those looked very different. Is that true or not true?
You have to wait for the next talk.
Gotcha. All right. Thank you.
And I will tell you.
Thanks, Mark. Denis, please. Denis, you're muted, mate.
Yeah, just I apologize because I missed up with the time. We are now UTC +2, and the meeting I thought it was UTC +1, so
my God.
I missed it. Sorry for that. But anyway, I'm catching up on the questions, and especially the one from Boturner. I think nowadays trying to map the conduction velocities is critical, in fact. I published a paper last year called Scaling in the Brain, where I put a framework for that. But it has been shown, for instance, that in patients in a vegetative state, they use mainly long fibers with high conduction velocities. Mm-hmm. And that's probably why they lose consciousness. On the other hand, depressed people, they feel slow, they feel heavy. But we know today that there is overactivation of the silent network and in some part of the default mode network where the fibers are very thin. Mm-hmm. So basically the conduction is much smaller. Mm-hmm. So what's quite interesting, if we have a bridge with physics, when the action potentials flow from fast fibers to slow fibers, of course transiting through activation nodes, there is also a change in the frequency of the action potentials. So we have a redshift. Basically, the frequency has to go down when using slow conducting fibers. I think this is completely new. So with some colleagues, we're now trying to convert the axon diameter to G ratio, but we need also to have an estimation of the myelin thickness, maybe from MTC, to get maps of conduction velocities. Mm-hmm. That's all. Yeah.
Yeah, but that's very interesting, Denis. So cool.
Thanks, Denis. Amy, again, please.
Yeah, just some of the conversations were making me think. I listened to a really interesting talk from someone who, is really doing myelin stuff the other day, and he was talking about, adaptive myelination and how myelin is very dynamic, and the thickness is changing, it changes along the axon's length, and yeah, I was just wondering, how does that kind of relate to the stuff that you were showing today, and your thoughts on that?
So yeah, so that's a good question. So what I think is that when you have a change of the myelin, so it's like, what is the function of myelin? So now I've just been talking about we're measuring from A to B, and we don't look at what is happening along it. But there are axon diameter variations, there's changes of the myelin, so that's why we also have a distribution of fibers. So you can see this P1 peak that I have, they are not coming all at the same time, even that it's the same region we are stimulating at exactly the same time. Still, we have a distribution coming out in the target area. So that suggests that something is flexible. And looking at the brain developed over many years, it makes sense. If it was very precise and it need to be precise all the time, then we would be super sensitive to small changes. Therefore, I think that is what I'm also saying, at the organization of the brain, at least in the white matter, what I see here, it is not complex because it makes no sense. And the same thing is in how they're conducting and the organization. It's a robust system. Otherwise, we will have many more problems that we already have now. Yeah, whatever. So, good question. I think it's a robust system that can handle or solve this distribution of things that are happening along the communication length.
Do you think that changes over lifetime as well? In that, like, when you're younger and you need more flexibility for learning, like, is it more of a, yeah, are there any lifetime effects,
No, I'm still getting smarter and smarter and smarter.
Good to hear.
I don't know, Amy.
You're an expert on the waiting room button. All right. I'm conscious of the time because people may need to get to a 4:30. So if you're still online and you'd like to turn your camera on one more time, no pressure. We can just show people, or show Tim, I'm sorry, that we're still here. Tim, I really enjoyed the talk, mate. I love the videos. There's things I'd never seen before. And I know they're going to make the recording available so people can look. And when you took, I could hear the pride in your voice about the visualization. You said, this is cool. I agree, it was cool. I think I saw it on either LinkedIn or Blue Sky, but that was awesome. So on behalf of all of us, another round of applause. Thank you so much.
Thank you.
Okay, just a couple more announcements then. Amy put a pitch for a PhD in the chat. Please have a look at that. I'm going to do the same here, prompted by that. And then that's for somebody who might want to look at the prostate. And we have next month, Corey Barron, who sent his apology, he had to go and teach. Otherwise, I would have asked him to, tell us about his talk. But he's going to talk next month. But then, of course, we get into ISMRM, then we get into summer. So we're really looking at dates. I'm just going to put them here you can read them there. And so if you're sitting here and think, I'd love to present, please drop Tim or me a message. I've already tapped up a couple of people who had previously expressed interest and we just hadn't got around to them. So if that's you, if you look at that and think I'd love to present, then we'd love to have you. Any of the announcements? No? Okay, well, we'll see you next month then for Corey's talk. And thanks very much, everyone. See you soon.